Thank you very much. So my name is Chloé Berton. I'm a postdoc at IMT Atlantic in the cyber health team, and I come from a background of cybersecurity, and I did my PhD on the security of data stored in DNA molecules. And to do so, I was under the supervision of Gwynou Coatrieux, so at the Latim Intern at IMT Atlantic. And so the work that I will present is a collaborative effort between people coming from the cybersecurity world, so Gwynou Coatrieux and myself, and people coming from the molecular biology and bioinformatics side with Dominique Lavenier and Julien Leblanc. So I will present our work, which is a risk analysis of the DNA data storage chain. So I think I will skip the context, but as you know, with the exponential increase in digital data production, DNA data storage is a promising alternative due to its density, its durability, and its storage energetical cost. So a single strand of DNA can be seen as a sequence of nucleotides, A, C, G, and T. And for this presentation, I will use the classical DNA data storage chain that begins with the encoding of binary data into digital DNA sequences with 4 bases A, C, T, and G. These DNA sequences will then be synthesized and stored safely. And to recover data, DNA molecules are first amplified and sequenced, and then post-processed with bioinformatics algorithms and decoded back to binary. And so, as this technology is still in development, there is an opportunity to secure it from the outset, meaning before deploying it into everyday applications. So I will now define how security is typically done for information systems. So security requirements are usually divided in 4 categories: confidentiality, availability, integrity, and traceability. And to deploy security in a company, a head of information system security, oversees the deployment and follows a plan-do-check-act procedure to offer constant improvement in data protection. And risk analysis is also conducted to first identify the threats on the information system and also to identify the specific security requirements and objectives to be met. So a risk analysis usually consists in 4 steps, which are detailed here. The first step is a context study, which establishes a global scenario to characterize the information system, so with several actors, assets, and data flows. Then sources of risk are identified, so those are the possible attackers and their objectives. The third step is the risk assessment, which evaluates the causes, the probability and impact of a risk to define its severity. And the last step is the risk management and treatment, which consists in implementing security tools and countermeasures to meet the security needs. For example, to ensure data confidentiality, a popular countermeasure is encryption. And so several risk analyses are possible, and we have chosen the STRIDE analysis from Microsoft for its ease of use and comprehension, as well as the wide range of threats that it covers. So let us define what a STRIDE risk analysis is. So each letter of the word STRIDE corresponds to a threat, itself associated with a violation of a security property that we wish to achieve in the system. For example, modifying or degrading data stored in DNA corresponds to the tampering threat that's associated with the integrity property. And as with all risk analysis, to identify threats in the data storage chain, we first did a context study and defined a use case for this chain. So what's a use case? In our use case, a client, a customer, wants to store several of their users' data in DNA molecules. So to do so, the customer will interact with several service providers that provide 3 different DNA functionalities that are storing data into DNA, archiving this DNA, and recovering data from the DNA. And so in this use case that I will show you, in blue arrows, digital data will be presented. Green arrows correspond to DNA, and red arrows correspond to reading instructions. So here's the scenario. To sum it up, when a client wishes to store their users' data, they send a request to a service provider that will encode digital data into DNA. To do so, they employ several specialists to do the encoding, special experts in synthetic biology to encode the data into DNA, and also to prepare the DNA for long-term storage. Then the DNA will be sent to a second service provider that will archive the data, and reading instructions will be sent back to the client. And so years later, when the client wishes to access the data of several of their users, the client will send a request to a service provider 3, along with reading instructions, and the service provider will get data from service provider 2, who does the archiving, and service provider 3 will do the sequencing and decoding of DNA to retrieve the data. So based on this use case, we have identified several sources of risk that are attackers with their objectives and motivations, as well as resources. So here are several sources of risk. Here is an overview. As we can see, we're talking about concurrent companies that want to obtain strategic advantage, or state services who have the means to threaten the system's security. And we also find specific sources of risk, such as a cyber bioterrorist who adds DNA that can lead to the creation of viruses. So as we see, the sources of risk are diverse, including some that are specific to the storage medium that is DNA. So let us now go through each step of the DNA data storage chain and identify security threats on them. First, there are some security threats that are present at all steps of the chain. First, they concern information disclosure, so data leaks. They are often done by an employee of a service provider. So I provided an example here. An employee can stealthily do some data theft, for example, at the sequencing step. When sequencing DNA, an employee can copy sequencing results, leading to data leaks. It's also possible for an employee at the synthesis step to mix the DNA obtained by synthesizing the data of several customers. Thus, when the customer 1 will retrieve their data, they will also find some data from customer 2, so leading to a security breach. So it's important to note that even if some of these attacks concern only small amounts of DNA with a density of up to 10 to the power of 18 bytes per gram, such data leaks can cause catastrophic damage. Another type of threat that's present at all steps is the elevation of privilege. Indeed, an attacker, if he gets access to the inside of the system, for example, a hacker can install some malware, modify encoding parameters, so it's a more general attack. Let us now see the the threats at the encoding step. So encoding is carried out by an employee of the service provider one. He receives the data and from the customer and organizes it to prepare it for the synthesis. So only one type of threat has been identified at this step. Here the aim of the attacker is to sabotage the encoding, to harm the customer or the service provider. So the employee can do a wide range of threats, altering encoding parameters, indexing parameters, adding parasitic DNA sequences. So here, the attacker takes advantage of his privileged access, and as DNA is a long-term storage medium, and DNA will only be read when needed, this attack can be detected very late, so have long-term consequences. Now, at the synthesis step, we have identified several threats. The first one is a bioterrorist threat. A hacker can add to the DNA file system some corrupted files that, when synthesized, will create some biological viruses. Another published attack consists in spying on a DNA synthesizer by placing a smartphone near the synthesizer, and by recognizing the specific noise patterns, an attacker can guess which nucleotides are being synthesized and thus steal some data. Other attacks include distributed denial of service attack, which means overwhelming a synthesis machine by sending many requests to hinder its activity. Now, at the archiving step, it's possible to tamper with DNA, with several attacks that depend on the level of access that an attacker has. It's possible to degrade DNA by modifying storage conditions, such as temperature or humidity. If an attacker has a more direct access to DNA, he can degrade it by directly influencing the DNA pool. It's also possible for an attacker to add a lot of non-coding DNA to a DNA pool to drown the data encoding DNA, and thus make it difficult for a service provider to retrieve the data. Another attack relies on repudiation. Here we here an attacker has direct access to the DNA and will steal part of a DNA pool, and then to hide the theft, we can imagine that an attacker can do a PCR to amplify the remaining DNA, and then replace the DNA in its protected state, thus hiding his theft. Yes, and the last step is DNA reading. Here, a wide range of attacks are possible. As I've said, in the last slide, it's possible to alterate DNA in a wide range of attacks. It's also possible to exploit the variable yield of sequencing by stealing some DNA and blaming it on a poor yield on the sequencing part. Several attacks also concern information disclosure. As with multiplex sequencing, data of several users are sequenced at the same time. A phenomenon can occur that's called index hopping, in which data of one user can be found in another user's file, leading to data leaks. In 2021, an article also showed that it's possible to recover the flow cells that's been used for sequencing, and that's in the trash in a lab, and to recover some DNA that encodes data, and thus recover some sensible data. And finally, a denial of service attack that's also been published in 2017 shows that it's possible to encode malware into DNA that, when read after sequencing, will cause problem to the computer it's been read on, as it contains a digital virus. So as we've seen, the threats are very diverse, and some are specific to the medium that is DNA. So there's a really strong need to secure the DNA data storage chain. So I will now show you a quick state-of-the-art of security solutions concerning the 4 properties of availability. First, it's possible to use, for example, error-correcting codes. Concerning the property of integrity, we can use hash functions or secure and have more control over what we're synthesizing by scanning DNA sequences before synthesis, or even doing a partial sequencing after synthesis to verify. Concerning the traceability property, it's possible to use digital watermarking, which is similar to what's called in biology DNA barcoding. And finally, for confidentiality, it's possible to use encryption, but these are digital tools that protect the digital medium, but are not specific to the DNA data storage chain. So there is an interest in developing a solution that's native to DNA data storage to take advantage and to really consider the medium's specific features. So I will show you some security proposals that do not simply rely on digital algorithms and are based on DNA. First, to ensure the traceability of living organisms, DNA can be watermarked by modifying some bases without changing the gene expression, which is a property that's specific to living organisms. Then steganography tools ensure data confidentiality by hiding data encoding DNA in existing media. So this greatly limits the density of stored information. And finally, DNA cryptography is a field in which encryption techniques are presented in the biological domain. So they fall into 2 classes, either with very limited density or are theoretical proposals that need an experimental validation, and that could be difficult to apply for DNA data storage. So thus, the problematic of long-term security of the DNA data storage is still open, and there is an opportunity to secure it now from the outset, before the technology is really normalized. So to conclude, the DNA data storage chain, as I've shown you, is vulnerable at all steps of the chain and to many threats, including some that are specific to the storage medium. And the sources of risk are varied, from concurrent companies to government services, as well as new threats, such as cyber bioterrorists. And the current risk management solutions to secure this chain are mostly digital algorithms that do not consider the specificities of DNA data storage. And so, as I've said, there is an opportunity to build native security solutions for DNA data storage. So thank you.
Are there any questions?
Thank you so much. This is some really interesting stuff that I just haven't thought that much about. I have I was curious about when you mentioned that you can encode like a computer virus and then it causes problems when it's read. Can you just explain a little bit more, like how that would happen?
Of course. So it's a published attack by Peter Ney in 2017, and the principle is that what's encoded into DNA is some shell code, so some informatics code that's encoded into DNA, sent to a sequencing service provider that does the sequencing, and when they read the FASTQ file, I think it causes an exploit in the computer, and if I remember correctly, takes control of the computer. So it's when digital files are processed. I don't know if I'm explaining it right, but.
So just a follow-up question is, if it's from the FASTQ file, the FASTQ files I think usually contain like a quality score from the sequencer, and I was just wondering, the person who put the virus in there can't control the quality score that's going to come out of the sequencer, so I was wondering if the quality score would throw off anything that's encoded in there.
I don't think the quality score is part of the shellcode. I think it's additional information that's in the file. I think I would have, so the reference is the first one. Maybe I would have to go back to it, maybe to explain more in detail how the computer exploit works in details.
Thank you.
Sorry. Thank you.
Any other question? No? Okay, I think we'll move on with the second talk. Chloe, this one was more on the threats and the security threats. The second is more focused on solutions. So that's why we thought it would be interesting to have the two talks.